The overall goal of this procedure is to fabricate PEG-based inverted colloidal crystal scaffolds with an extracellular matrix coating for liver tissue engineering applications.
Liver tissue engineering is an exciting field for tissue engineering and regenerative medicine. We are creating 3D engineering scaffolds that allow liver cells to maintain their function for a long time.
The implications of this technique extend toward drug testing, as liver cells cultured with this system may retain their function longer and better respond to the drug.
Though this method can provide insight into liver tissue engineering, it can also be applied to other systems, such as those using stem cells.
Demonstrating the procedure will be Hitomi, a graduate student from my laboratory.
To prepare the microbead molds, first cut the tips from 0.2 milliliter boil-proof microcentrifuge tubes at the 40 microliter level. Use waterproof glue to adhere the tops of the cut tubes to glass microscope cover slips and then glue around the molds. Next, replace the water in a vial of polystyrene microspheres with 70% ethanol. Place the sphere solution into an ultrasonic bath and sonicate the mixture at 30 watts for 3 minutes to loosen the aggregated spheres. Once the microspheres have been prepared, pipette 100 microliters of ethanol into each of the molds. Then, cut 4 millimeters off of the top of a 200 microliter pipette tip and use it to pipette 25